And so this is where we're at. Welcome, and like I said, we of course wanted to do a class day. Here is another set of internships that students can sign up for. These are in the summer here at USM and out in surrounding areas. And the Beer Lab, and if you haven't heard of the Beer Lab, it's a opportunity on campus and surrounding. And it's a paid internship. It's a really good experience, and lots of students do it. Here's a great example. This is Katie Knox doing it, and she is very enthusiastic pre-med student. So she's not interested in brewing beer, but she's interested in getting some hands-on skills. And Katie went on to do other internships. So she did one of the internships down at Maine Health Institute for Research, like that. So, she was more ready for that next step. And so those are happening, and this is something just unique to USM. So it's not particularly competitive. There's a little competition to get in. You have to apply. But it's a really great starting point like that. And so I'm happy to talk about that. And the person who's really in charge of that is Samantha White, and Sam is a fantastic person who can also do that. So what should you do this week? And we'll talk through what we're going to do this week, okay? So a week ago, we didn't have class because USM opened at 10, right? So I made this snow day lecture, and in it, I made a drawing assignment. And it was just drawings that we would have done in class. But I also want to continuously say, let's draw, let's draw, let's draw, and encourage you to draw in class and encourage you to draw in assignments. So I made those, and those pictures of those are due today, like that, okay? And if we have a snow day, I mentioned, or an interruption like that, I make that as if you're gonna watch it pretty soon, like that. So it's just as important as our class meeting, and I just move on, from there, like that, because we have this tech, like that, right? And so this is just an example, not of drawings from this course, but just other drawings in a drawing assignment students have done in the past, right? And I'm going to make a comment about that, and that is when you draw something out, okay, that's what you're going to remember. You're going to remember that on an exam. And I'm not talking about being good, fancy artists. Sometimes students that are the neatest and have the neatest penmanship stumble on this because they put too much time in crafting it and not making it a learning tool. And the key concept for it as a learning tool is you're going to remember that image, and you should try to spread it out. So your mind can move to different points in that and see it and see the words like that. So you're doing this for yourself, right? And I mentioned before, I think, that I grew up in a little farming town in Minnesota, and it was very weak school system, focus on farming. That's where you will learn. Get out, do some farming. I have a bunch, had several classmates that quit high school when they turned 16 because they were farmers, and that they had to legally go to school till they were 16. They have no interest in it. So very weak, and then I went off to this really strong college, and I had to really scramble to learn. And so I'm sensitive to that as part of the college experience, is to try to figure out how you, uniquely as a person, learn your best, right? And it turns out that I think this setting is a really great one to learn about your learning skills and how to strengthen them like that, okay? So here's just a little thing, and you can look back on it. You don't have to read it right now. You can look back at the recording. But the education research shows that in STEM learning, this is the way to learn STEM. You don't just pick up a STEM book and read it, page 2, page 3. You don't just drive down the highway listening to the recording. You actually get it out and make it physical, and make it big, make a model like that. And there's a whole bunch of reasons why that's strong and very transferable, and it improves your learning in general. I want to just show you this. This is a company called Sketchy. You don't have to look at this. It's an example of their website. And Sketchy makes a different kind of drawing, not what we're doing in our course, but a different kind of drawing for medical students. And so this is not a course in medical school. I'm not encouraging you to do this. But Sketchy does that, and they're having a lot of success. And that other website is where they've started to try to do this same strategy for college classes, not medical students. And this is what Sketchy looks like, it'll be a drawing like this. And so this is a drawing of a cartoon where kids are trick-or-treating, and there's some isolated things happening. And there are about 30 different isolated things in this one sketch. And the medical student does this. They say, trick or treat for sulfa drugs. And those are 30 facts that the doctor has to remember about sulfa drugs for their career. So the doctor, for the rest of their life, will see kids trick-or-treating, and they will think, trick-or-treat for sulfa drugs, and they'll remember that. Because anytime sulfa drugs come up in their career, they have to remember that. I have a son-in-law who's a resident in child psychiatry, and when he was in med school, at the end of med school, he had to pass step 2 exam. And he was studying for it, and that's where I heard of this. This is the only place I heard of this, because my son-in-law used that. And I said, how many of these do you have studying for step 2? And he said, 250. So we're not doing that. We're not doing that, but it's an example of how, in fact, we evolved to learn in the real world. And we have to be mindful of that when we start talking about the molecular world, right? And we need to zoom it up to make it physical, visible, memorable, have cause and effect relationships like they happen in the real world, okay? So just a little of that. So another drawing a student did, write some notes, that's great. You know, I've described how you make it big and have flashcards with notes on them, stuff like that. Here's a drawing, you see it's different, you know, different people, different drawing styles, that's fine. And, you know, can you remember that? Can that student remember that? It's all for you, really. Another drawing like that, okay? So drawing. So we had some drawings in that snow day lecture. I drew drawings fast. I'm sloppy left-hander, you know. I apologize for that, you know. It's not a good model, you know. If you had somebody with good penmanship, you'd be like, oh man, I really can learn from his drawings, right? And I apologize for that, but we have to get those in today. Just pictures of what happened last week on the snow day. So then we'll do a quiz 1 redo at the end of the class today based on that quiz 1 answer key video. And then I also have quiz 2. So I'll give quiz 2 back for you, and I will post a quiz 2 answer key video. And there's not an assignment with it. I'm not going to do this again, but you really want to look through it, even if you got all those points. And students did great in general on the second quiz. Great. I feel really good about it, but I still make the answer key video because we want to loop through it and keep trying to learn. Then we're talking about chapter 7 today. That's the last chapter I can put on electronic reserve. You should have the book. The next chapter we go to is chapter 16. And I've been leaning into that chapter from the first day of class. I've talked about that. FOXO is in chapter 16. RAS is in chapter 16. So we're going for it. Then we also have an assignment due this Thursday. There is this Wisconsin to Boston video assignment, and it's got drawings, and it's about that retrovirus, and it's about the normal retroviral life cycle, and it's about the 2 points that really matter to us, and one is it's got to copy its RNA into DNA when it infects a cell, and it uses a retroviral genome produced retroviral protein called reverse transcriptase, and that's a powerful tool. Used as a tool. We'll use it in the lab class. I mentioned that. Excuse me for my sniffling nose, but I was running around getting that quiz. And then the other one is part of the replication process that retrovirus has to insert that DNA into the host genome, and it might accidentally insert it next to an oncogene. It might pick up a host gene. And when it does, it becomes what we'll call an oncogenic retrovirus. Okay? And our book talks about that throughout, uses that concept throughout. There's some famous oncogenes, and we need to go from their origin of their discovery in places like Wisconsin to their further study in places like Boston, where our author moved to Boston, and he did not study retroviruses ever again. He studied RAS, and he found it in mice, and he studied it in mice, like that, okay? Then we have another assignment that will be due next Tuesday, and it's straight out of chapter 7, but I put chapter 7 topic into a package like that. So one of the chunky things in chapter 7 is methods of how scientists study and identify transcription factors. And it is in chapter 7, but I consider it to be kind of a little bit disorganized, you know, because there's a way to put some together, and I just put 5 together. They're mostly represented in chapter 7, so that's when we can take a chunk of 7 and make it into in this case, it's a writing assignment. So try something different, not a drawing assignment, but in this case, I have 6 questions that you have to answer as part of the video. And there are questions that have been on the exams. 2-point questions, so we should have some of that, right? Practice learning and thinking about how we learn to get ready for the exam, okay? So, and so I kind of have these out of order, but chapter 7 and then on to chapter 16. And here we are on Tuesday. It's 3 weeks till our first exam. Our first exam will cover chapter 1, 7, and 16, and everything else. So the videos and the assignments and the quizzes, all that will be on exam 1, 3 weeks from today. So we have 3 weeks. This week, it'll be chapter 7, and then 2 weeks of chapter 16, and our exam, okay? So I believe that's where we're heading. I hope I looked at the calendar correctly, and that's what we're doing, okay? And you can double check that on the syllabus, okay? So chapter 7. So the videos that we started the course with, the muscle differentiation videos, those are chapter 7 material and more, okay? So we really want to learn those. And so here's some more things as we get into chapter 7, some more things that I just wanted to remind you of and then add to. So the first thing is from the assignment of drawings from the snow day lecture a week ago is the concept of cis mutations and transmutations. And so those are terms from genetics. They're in that video, and they're super important because from them we get the term cis regulatory region. So a gene is physical, made of DNA, composed of 2 parts. One part is the transcription unit. It's going to be the product, the output of the gene. That makes sense. We use portions, just like computers. The memory of a computer is divided up into files, and you open the file you want to be using at that time. So then the second part of the gene is the regulation of the gene. And that's we now call cis regulatory region. Because we know the MyoD gene affects the myogenin gene, but it does it through a trans action. On the other hand, to be part of the myogenin gene, it has to be physically connected. So the cis regulatory region means physically connected. We'll talk about it. The enhancer can be a good distance away from the transcription unit, but it has to be physically connected. Okay? So that's that first thing. And then also that a single transcription factor is always present as thousands of molecules, and it likely has multiple target genes. MyoD probably activates multiple genes. We just focus on myogenin, right, to give a nice clean example. Myogenin definitely has lots of target genes, lots of muscle genes. Boom, we've got to activate a lot of stuff. We've talked some about the estrogen receptor. We'll talk about it more this week because it comes up big time in chapter 7 and then right at the beginning of chapter 16, our next chapter. The estrogen receptor has hundreds of target genes, right? It's going to turn on everything that's going to happen during puberty, like that in vertebrate females. So that's great. Then another thing is this idea of feedbacks of gene expression, and we want to be paying attention to that. And that's where the idea of, the transcription factor could get turned on and off. That's in a loop. So all transcription factors are proteins. That must mean they have a gene. So there's a MyoD gene, and I wrote about that and drew about that. The MyoD gene gets expressed. The RNA is mRNA. It's going to go out of the nucleus to the cytoplasm, bind to ribosomes, and the MyoD protein gets made. The MyoD protein has to get back into the nucleus to do its job. And that's true for every transcription factor. They get made outside of the nucleus, and they get back in. They got to get back in. And we've seen FOXO. FOXO's regulated. We're not letting you back in. Like that. Okay? And there are other transcription factors that are regulated in a variety of ways. And that allows for great complexity of gene expression. We get all these genes from our parents, and we don't use them all at once. We don't use the estrogen receptor target genes for years. They're there all throughout childhood and you know, youth of every vertebrate. They're there and they're not activated because there's a loop. Yes, a receptor gets made, but to activate the genes, you need that hormone. So that idea of feedback loops is just gigantically important, and we're learning about them in part by learning how transcription factors get turned on and off. So we got that big picture list that we're building that includes so far MyoD and myogenin, FOXO, estrogen receptor, FOXO, and now ALK is on that list, like that. So we'll keep adding to that. So here's the last thing, and I haven't described that. We've talked about a closely related thing, and I just want to add to that. So we talked about FOXO. So we're going to use FOXO as an example. And we've talked about how FOXO could be inside the nucleus or outside. So then separate from that, we introduced that glowing protein, GFP. And we've talked about how you could make a transgene and we could put the myogenin cis-regulatory DNA from a mouse together with the GFP transcription unit from the jellyfish and put that transgene into a mouse, and the mouse's muscle will glow. So this FOXO story has some similarities to it. We said, let's try to label the FOXO protein, and either the nucleus, the nuclei in the pellet would glow, or the liquid would glow, depending on where the FOXO protein is located. So what we got to add is this. When we talked about the myogenin expression, we were making myogenin in the muscles. That means we're making the GFP protein in the muscles, and it just floats around in the cell. So the whole cell glows, the whole muscle glows. Because it's this foreign protein, it doesn't have any special place it's supposed to be. It just floats around and the whole cell glows. This is not that example. This is, we want the protein to glow. So I just want to describe this. It's just a slightly different gene design, okay? So let's just draw this out. And so what we want to do is we want to use that jellyfish GFP gene. And remember, I say you're going to always draw and label the parts, but I do it quick. And so this is the jellyfish GFP gene, and we know we're going to want it, you know, I'm going to put this, you know, kind of way out here, but that's what we think we want, right? We want that part that can glow, all right? Now I want to add to this, in the transcription, a portion of that is the coding region that's going to really make the protein glow. That's going to make the glowing protein, right? So the transcription unit is bigger, right? So then we want that FOXO protein to glow. So here's the FOXO gene. Again, you know, label it up, and it's FOXO, and we're working with a mouse, okay? And here's the coding region, right, for the FOXO gene, like that. So in this case, we don't want to swap the cis-regulatory regions. In this case, what we're going to do is we're going to take that coding region and we're going to insert it right into the end of the FOXO coding region. So we'll have one long coding region that makes FOXO protein and then goes smoothly into GFP protein. And we can draw that, what that protein would look like. When that protein gets made, we literally have a portion of it being the mouse FOXO protein and a portion of it being the jellyfish GFP. Now, you might quickly say, would that even function? And the answer is, it doesn't always. Sometimes you kind of wreck the FOXO protein. So you have to make multiple examples of this to try and get one to work. But what happens then is, remember, the GFP would just float around in the cell. But now it's physically connected to FOXO. And if FOXO's in the nucleus, the GFP's in the nucleus, the glow is in the nucleus. the example I always give is, what can we say about my feet if I say my head is in the room? And the answer is, my feet are in the room, because they're physically connected. So that's just, we've talked about that idea. Let's make glowing foxhole. This is how we have to make it, and it's a great example of a tool being used, and it's a great example for learning because you see that, right? We want a glowing foxo protein. What will we manipulate? The coding region, right? We want to change the product, okay? Any questions about that? And that goes back to, woo! I don't know if I have this again, but this is making transgenic, and people call that a chimeric protein, because a chimera is from olden days, like in ancient Greece, where they had like a horse body and a human, torso on top of that old horse, like that, or like a horse with wings, Chimeric means kind of a monster hybrid, and so we make that, okay? The mouse is normal. It's got the regular FOXO gene. We've added an extra gene, like that. The same is true for that glowing mouse in our muscle videos. It's got all the genes. It's got a normal myogenic gene. We just added this extra gene, like that, okay? All right, so that, and that's just building more about what we're getting out of chapter 7, because we've been leaning toward this. Chapter 7 is like an introduction to molecular biology, right? So it's a good place to go and get busy, right? Like that, okay? So the next thing I want to say is that it comes up in the drawing for the snow day lectures. When we say gene is composed of two parts, the transcription unit, the cis-regulatory region, the transcription unit is going to be the product, and the cis-regulatory region is the regulation of its expression. And you just memorize this. The cis-regulatory region determines where, when, and how much, because that's all it ever does in any cell on our planet, and it always does that. Where are we going to express it? Well, in the muscles. When? When muscles start to form. How much? Could be more or less, like that. I think it's pretty quickly clear that the where matters because of genomic equivalence. We got all the genes. Where are you going to use the insulin gene? Cis-regulatory region determines that. And then the when also. Are you going to use it in a baby? Going to use it during puberty? Going to use it throughout the life? Now, how much is just a little confusing to students, and so I wrote that big here. How much, okay? And you would say this, you'd say how active, how strong is the cis-regulatory region? So how much gene expression? Because it varies from gene to gene, and some genes get expressed at a very high level. And what that means is the transcription factor is making the gene get expressed, the RNA polymerase go, 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 like a really busy airport where the RNA polymerase is landing on the transcription unit, and here comes another one, and another one, and another one. So very strong, very active, okay? So to remember that, how much story, we should have an example, okay? And, before we do that, just to mention this, there are really two ways of measuring how much is being expressed. And we look at those airplanes landing. Are there lots of RNA polymerases landing or not so many? And the two ways are called a nuclear run-on assay and a grow-seq assay. And they're really the same thing. The only difference is nuclear run-ons analyze one gene and a grow-seq analyzes all the genes at once. And so we're really just going to learn a little bit about the nuclear run-on method, and we're not going to learn about grow-seq. But we should mention this sometimes in our course, where some of the methods we'll learn about have been turned into big data methods. So that's very true in biology, modern molecular biology is big data. Why should we look at one if we Shouldn't we just get them all? Let's get them all. And so this is an example of that. So you can learn those too, but I would ask you about a nuclear run-on. Like, what's a nuclear run-on do? Nuclear run-on measures how much gene expression is that doing. Quick side note, why can't we just look at the RNA levels in the cell? And the answer is because some of the RNA is getting degraded. And the rate that it gets broken down affects the amount. So if we want to say how much, we have to really look at the activity on the gene, because the RNA is not a good predictor of how active the gene is. Okay, so a nuclear run-on. So a nuclear run-on is this simple. Remember this? We're going to grind these cells up and isolate pellets of nuclei. And that's why it's called a nuclear run-on. Is the first step in a nuclear run-on is to make nuclei. Now that we have isolated nuclei, we don't have to worry about the cell membrane. We can add things to the nuclei, and they just go into the nucleus. And what we're going to add, we'll isolate some nuclei, and we'll add some radioactive AGCT, or U, not T, but U, because that's what's used in RNA. And that will measure that new production of RNA. And we have the nuclei, we isolate them always in the cold, so they're essentially living, active, but paused. And then we'll add the radioactivity, and we just warm it up to the body temperature, and we just wait a minute, and then we analyze those radioactive tags. How much do we get from, the myogenin gene, like that? Yeah, that's an indication of how active. And so a nuclear run-on is that simple. Is we want to measure how rapidly that gene is getting transcribed, how much. So now that we know how to measure it, once in a while we'll have an example, and we'll say, this is expressed, and I say that here. This is a strong system regulatory region. That's the term they use. When they say strong, it means a lot. It's going to make a lot, okay? All right, so let's now use a practical example of that. And here it is in the oncogenic retroviruses. So remember, there were a couple dozen of these found in non-human species, and one of them is called RAS. And we've already learned that the problem with RAS is always it has a mutation in the coding region. It's making the product that can't turn itself off. So let's just back up and say, the thinking is, the interpretation is, when that first retrovirus accidentally picked up RAS, it probably wasn't a problem right away. But viruses don't do a good job copying. They make a lot of viruses really fast, and they make a lot of mistakes. So the thinking is that pretty quickly, that virus that accidentally picked up a normal RAS got mutations, and one of those mutations was in the coding region, and it decreased the activity. So just think about that, right? That infects a cell in a healthy mouse, we'll say, and the mouse has normal RAS and normal RAS signaling, but we've just added an extra RAS, and it's wrong, and if it gets used, it doesn't turn itself off very well. And that's all it takes. This is like an Achilles heel in that pathway. Just go, because RAS is often on a growth pathway. It's often stimulating the cell cycle. Chapter 17, the fourth chapter after the first exam, is the cell cycle. How do these transcription factors and so forth, how do they regulate the cell cycle? So that's what's happening with mutant RAS. So there's another famous oncogene that got identified here, and we're going to look at, in our whole semester, about 7 of these total that were originally from those oncogenic retroviruses. So the book keeps mentioning those, but again, we really left the virus behind and studied the genes in a normal body and in a body with cancer. And the 2 we're going to learn about for our first exam are just RAS, and this other one, MYC, M-Y-C. So MYC was discovered as a part of an oncogenic retrovirus, and this is the thing for our course about MYC. MYC, when it's in an oncogenic retrovirus, is normal MYC. Remember, RAS gets to be a mutant RAS protein. But MYC is normal MYC. And the problem with MYC in this oncogenic retrovirus is viruses have very, very strong cis-regulatory regions. They infect a cell and they are just taken over. How do they take over? They express their genes just unbelievably high. So we say that for any virus, it has a strong cis-regulatory region. And when the cell actually picks up MYC, MYC gets overexpressed to just a tremendous degree. And even though it's normal MYC, it causes cancer because it's too much. And after the first exam, so later in our course, we'll learn about some of those oncogenes, and we'll learn more about why is that a problem? You know, why is it a problem if there's too much MYC? It's normal MYC, it's too much. We'll learn about that. For our first example, we want to focus on just, oh, it's another one, and it's MYC. And see, we can compare and contrast RAS and MYC, right? Goes back to our first definition of a gene. The mutations in RAS that cause cancer are always in the transcription unit. Mutations that cause MYC always involve changing the cis-regulatory region. So it makes too much, like that, always. Yep. So in one case, we make the wrong kind of RAS, and that's a problem. In the other case, we make too much of the normal MYC. So it's a great example of how much, okay? So RAS and MYC will be on our exam. MYC is a transcription factor. Great, like that. All right, so next thing. It's great to talk about RAS and MYC for our first exam because RAS is involved in this pathway, regulating gene expression, and sometimes one of the target genes is MYC. So let's express MYC. So another way that is great to introduce MYC is this, right? If we have a mutation and we don't turn off RAS, we'll make too much MYC, and too much MYC is a problem, like that, okay? Now, it's not always a target gene, but sometimes it is, and there are other target genes, right? The target genes are really, the target of RAS, are really the targets of this ELK transcription factor, because that's what RAS is doing. It's activating, it's phosphorylating ELK, and ELK is turning on its target genes, and sometimes one of the ELK target genes is MYC, like that. And so we could zoom in on here, right? Because we'd like to start drawing the MYC gene, right? Thinking about MYC and the cis-regulatory region. So let's just do that. Because you'll see that we pick on MYC because it has a very important cis-regulatory region. So here's our first time of drawing MYC. It makes a protein, so this is a coding gene. There's MYC, and of course it has a coding region in the transcription unit. It's got a promoter. And we'll learn MYC has multiple enhancers. And we just learned that one of the enhancers must be for binding ELK. ELK protein. And it would be that phosphorylated ELK that got activated through that RAS signaling pathway. Okay? So transcription regulation, a specific example that we're pointing ahead beyond the first exam. All right? When's an example of when this happens? And the answer is under every scab we ever had and will have. And that's, I mentioned that before. So under a scab, this pathway gets activated. And what are we trying to do? We're trying to heal. So our skin cells are always slowly dividing, rubbing off dead skin cells, staying healthy. But under a wound, we want to heal. So we're going to speed up the skin cell cycle. So this example could be one of your skin cells, totally normal, healthy skin cell under a scab. And I already mentioned that we'll learn about this growth factor, and it's this under the scab, it's called PDGF, platelet-derived growth factor. And you probably know platelets are involved in clotting. They're also involved in healing. Super satisfying example. So we'll dive into that some more. But it's important to put this in context so your mind can move to a drawing, and your drawing can be big old scab, and you're diving in under it to see what's happening to those skin cells, okay? So again, started talking about oncogenic retroviruses, and then we roll on to our normal body and what can go wrong with it can lead to cancer, okay? So how much, right? That oncogenic retrovirus and MIC is a great example because that retrovirus is just going to crank up the expression of MIC way too much and keep it going, like that, right? Okay? All right. So I wanted to loop back into some of that to kind of help us, get more of the snow day lecture drawings, but also take those a little bit further, take that example of the glowing, cell or protein a little bit further. So now we can start talking more about 7, and this is how we'll deal with all of Chapter 7. Chapter 7 really has 3 parts that we want to talk about. And our book talks about them, but not with this same simple organization. And the 3 parts are, everything we've talked about with regard to those clay models and genes and just what we just said. All that stuff is Chapter 7 stuff. And then there's these 2 more things we haven't talked about. The first thing we're going to talk about is this. So what does it mean when a transcription factor increases the gene's expression? What is it doing biochemically? What does MyoD do to turn on the myogenin gene? And the answer is it's somehow going to increase the rate of transcription. And that's done by RNA polymerase, so we're somehow increasing the rate of transcription. And different transcription factors do that in different ways. So I'm going to talk about that. And then this other part is going to be a homework assignment. We'll take care of that. And that's that drawing, that's that answering assignment where I say, you're going to go through those methods and then answer questions like they're from a quiz. And so that's due next Tuesday, I think, like that. So I'll post that, and it's this assignment, and it'll be in that package. And we'll think about it that way, because it's some of these methods that people use. And the book talks about those methods. So when you think about Chapter 7, and you try to tackle the book, tackle it with this kind of organization. But parts of it are helping us understand the stuff we've talked about so far. Then this new thing of what can these transcription factors do? What do they biochemically do? And then the third thing, some of the methods that used to study it. So what we're going to talk about for the rest of the time today and Thursday will be the second thing, okay? So if you look, at all of the transcription factors in a human, and there are about 2,000 of them, they all regulate transcription through a specific pathway, and some do it differently. And there are a variety of ways they regulate transcription. But there are really 3 major ones. And so we're going to learn about those 3. And the 3 major ones are just listed here, and we'll go through them. So how do transcription factors function? And the 3 major ways they function are, and we got to talk about what this means and give examples. They help us assemble the pre-initiation complex. They release what people call a paused or arrested RNA polymerase, and they remodel chromatin. So there are other ways they do it, but these are the 3. So, for example, in humans, it's estimated that 70% of our transcription factors regulate this second style. Okay? So here's the next thing to say about that. What they are regulating, for example, what MyoD regulates on the myogenin gene is called the rate limiting step. And so you have some familiarity with that term from chemistry. Chemistry will have a series of chemical reactions, and one of the reactions is the slowest, and that's called the rate limiting step. And if you want to increase the series of reactions to occur, you would target the rate limiting step. So I'll just show you this. I think this is here close. So to show you this first one. For this first example, they help assemble the pre-initiation complex. What's an example of a protein that does that? MyoD. So we can say right now, the rate-limiting step in the transcription of myogenin is assembling the pre-initiation complex, because that's what MyoD acts on. So just looking a little bit ahead to get that idea, and we'll go back to this rate-limiting step. And so the way I describe the rate-limiting step is like this. So if you ever go to like one of these county fairs, like Cumberland County Fair, there'll be these food booths, and they're open and you can see in them, and they're run by some volunteer organization or like a church group or some community group. So it's volunteers, and you can see them working. And so you'll get in line and you'll be waiting for your cheeseburger, and you can watch them making them. And some of these people, have worked in restaurants, but many of them are not. They're just helpers, and you can watch them, and you can see who the rate limiting step is. And it's like, we're gonna say, it could be the pickle lady, And that's the rate limiting step. You can see there are cooked burgers lined up in front of the pickle lady. And they're just waiting for her to get pickles on there. And if you get to the front of the line and you could say, hey, you guys might want to fry these burgers up faster, that's not going to help, right? There's just going to be more burgers waiting for the pickle lady. So that's what we're talking about. The pre-initiation complex, we'll see, it's a step pretty early, like that. The rate limiting step is something like getting the burgers fried. And the later steps would move, they're moving plenty fast. So if we want those guys to produce burgers faster, we tell the pickle lady, we have to get the pickle lady to do faster. And that's what these transcription factors are doing. The MyoD protein functions by increasing the rate limiting step in myogenin transcription. And the rate limiting step is this assembling pre-initiation complex. It's very much like it affects the pickle lady at that, yeah? Okay? And we'll see that some of the examples are early steps in the process of transcription, and some are later, like that. And so just to describe this, because it's pretty curious to see, this one, which is very common in our bodies, it releases an arrested polymerase. That means it's very late. This pre-initiation complex is set up, the RNA polymerase is there, the RNA polymerase is started, and it's going down the gene, and then it stops and sits there, like that. So we'll talk about an example of that, right? But back to the analogy of the burger shack at the fair, that's like the rate limiting step is the person who's handing the burgers out, and they don't let go of the plate. Because they're like, man, it's hot out today, right? And you're in line waiting, and there's this super chatty person who's handing out the burgers, and they just won't let go of them. And that's the rate limiting step. So for these genes, the rate limiting step is this releasing the arrested polymerase, a bizarrely late process, where the gene is really active, but then it gets stuck at the very ending, like that. And so that's what rate limiting steps are, and it's like the burger booth, because all this is a very physical, factual stuff. You're not gonna change it if you do something else. It's not gonna help by yelling at the pickle lady. It's the dude that's too chatty. Like, oh gosh, those Red Sox, dang it, they're so good this year, blah, like that, right? So that's what we're talking about in this. What is the rate limiting step in that gene? And that's what that transcription factor is going to modulate. And we usually think it's going to activate it, but there are repressors. It could slow it down, okay? These are going to be all examples of activators. So the next thing to think about that is this, okay? So it goes to the idea that when we draw a gene, this transcription factor we're typically talking about is a transcription factor that's bound to the enhancer, and it's cis. So here's the enhancer, and that's the part of the cis regulatory region that we'll really focus on in this introductory course. And we've got a protein like this, MyoD, okay? And it's going to increase the transcription of the myogenin gene, right? Like that, okay? So it's cis. And that turns out to be a really significant part of the story. It's got to be connected to the transcription unit. The promoter is always right next to the transcription unit, and it really just shows where we're going to start. This is where the RNA polymerase should land. So people still like to call this an enhancer for historical reasons. And the historical reasons are interesting to talk about. And that is, when they first found enhancers, they were curious because the enhancer was far away. Sometimes the enhancer could be like 10,000 nucleotides away. But as long as it's cis, everything would be fine. And so they found some facts. And one is, the enhancer location didn't matter as long as it's cis. It could be very far away. It could be at the other end of the gene. How does it work, right? They didn't know how it, affected transcription. Or you could take the enhancer and you could just flip it around. So in a sense, it's pointing the other way. Because some people thought maybe the enhancer, transcription factor would bind the enhancer and help kind of load things and have them slide on down. But it's not doing that because you can point it the other way, and it's not directional like that. So that's why people started calling this an enhancer, because it gave a kind of a simple statement of these experimental results, because that's always the way an enhancer works, is you could move it around and you could flip it, but as long as it's cis and it works. So what's going on there, and the most likely explanation is this, that we've simply tethered the transcription factor to the transcription unit. Like put it on a leash. And if something's on a leash, it doesn't matter if the leash is very long. So for example, I use this example. I'll say, if you're out like on a football field and there's a bitey dog running around, you're probably not gonna get bit, it's got a lot of area to cover. But if you put a bitey dog on a leash, you're gonna get bit in about 2 seconds. Because it's your area now. You'd say we increased the concentration of that bitey dog. And that's understood to be what's happening here. We've just increased the concentration of MyoD in this area. It's like running around on its leash, and it's going to bump into this part here. MyoD is going to increase the assembly of the prenatiation complex, and that's going to be RNA polymerase, and we'll learn proteins that bound here. And it's just going to help that get assembled, because it's like in the area. It's like go, go, go. There's a term that you'll appreciate about the dog and the MyoD that scientists that study transcription like to use, and they say a transcription factor increases the local molar concentration. So in the cell, there's lots of molecules of MyoD. There's a concentration of MyoD. In the football field, there's a concentration of the bitey dog, and the concentration is pretty low. Even though there are lots of MyoD molecules, the overall concentration there, all throughout the cell, it's pretty low. If we put it on a leash, we're concentrating it in a smaller area, and we're more likely to have it do its job there. So that's the concept. Why is it cis? Because it has to be physically tethered. That's what's going on. I should mention these letters don't matter at all. They could be A, G, C, T, or anything else. They just have to be a leash, like that, okay? And so that's how it's going to increase the transcription, or decrease it, is it's going to be tethered and it's going to be doing its action, okay? And I think with that, we got a couple minutes, we're going to wait for my alarm to go off. So that's that idea. Questions about that? So people like to call it an enhancer for that historic reason. but it also helps kind of illuminate when you hear the definition of an enhancer, works at a distance, any distance, as long as it cis, works in either orientation. That's what a leash is like, right? If you put a dog collar's leash on, collar on backwards, works just as fine, it's fine, right? Because all it's doing is it's concentrating that bitey dog near your leg or near the start transcription, okay? So that's that idea. So transcription factors often function by increasing this local molar concentration. And that's it, I put in quotes because it's a very unofficial term, but you get the concept. Molarity is a concentration, and we are concentrating that bitey dog in a smaller area. So something needed to increase the rate limiting step. Okay? All right, and so I just drew 2 of these examples up here just to give you an idea that you don't need to scramble to write that down, but just to give you the visual that we're talking still about something that's very physical. Remember on the muscle differentiation videos, I made the genes out of clay. And I said I made that because you can cut it up. And we talked about that. Also out of clay, because clay is floppy. And if you look back at the videos, all the clay is kind of curved to show, it's not like you see it in the book, a straight stick. It's floppy, like a leash, like a tether, okay? All right, so our examples will be this. So the first example, MyoD and the myogenin gene. And so you'd be able to draw this, and add this to our drawing, right? We could draw a myogenin gene, right? And there's the promoter, there's enhancer, there's MyoD binding. And what we can add to it is that MyoD is doing something here, and it's increasing the assembly of this pre-initiation complex. Okay? And so that's a good place to stop, because we're going to stop there. And, it's just going to add to that, and it's chapter 7 stuff. Okay? All right, and because our examples are chosen to illuminate the stuff that chapter 7 wants to talk about. So